Okay.
Hello, everyone, and welcome to phyloseminar.org. This is the first talk in a series in 3 talks on next-generation sequence evolution models. Please use the YouTube live comment box to ask questions. Today's speaker is Dr. Faruck Morcos. Dr. Morcos is an associate professor in biological sciences at the University of Texas at Dallas with affiliate appointments in bioengineering and physics. He directs the Evolutionary Information Lab, focusing on the intersection of biology, computation, information theory, and biological physics. His research develops methods to extract biological information from sequence and genomic data, creating models for molecular evolution, protein structure, function, and design. Recent work includes machine learning approaches to study protein sequence space and unified evolutionary models. His lab predicts mutation effects in protein families to design chimeric repressors, biosensors, and discover variants that could restore function in disease-related proteins. Dr. Morcos completed postdoctoral training at UCSD and Rice University, earned his PhD in computer science from Notre Dame, and holds an MS in electrical engineering from the Technical University of Munich. Welcome, Dr. Morcos, and thank you for participating.
Well, thanks a lot, Eric. I really appreciate the invitation. I'm kind of honored to be here. I saw the list of previous speakers, and I'm really glad that I have the chance also to present in this seminar. I think this is a great initiative from your side, and it's exciting to be here. Let me start just by giving a little bit of background. I know that this is kind of a seminar for people who already are interested in phylogenetics and evolution, but I still would like to go a little bit through some background for the benefit of general audiences. Okay, so yeah, thinking about some pioneer research in molecular evolution, we can go all the way back to the 60s, where Emil Zuckerkandl and Linus Pauling proposed a view on how to look at molecular evolution in a little bit more rational way, based on the idea of molecules as opposed to maybe only fossil record or morphology. And although they might not have been the first, they did have a very influential paper called Molecules as Documents of Evolutionary History. And then they actually think about a kind of preservation principle that connects kind of the change and through evolution, the change of molecules through evolution, and that kind of tracking of change in molecules could maybe tell us something about kind of relationships between organisms, and maybe use it as a molecular clock that it's hard to track in different formats. And they claim that this could have been an important way to study evolution. This was in the 60s, and it was a kind of thought experiment more than kind of a scientific paper. So they thought about, okay, there's possibilities. What should we use? Should we use DNA, RNA, or maybe polypeptides like proteins? And the key argument and the conceptual advancement was the idea that, well, if we compare homologous protein or polypeptide chains, maybe we can actually, just like we do in phylogenetics, well, we can determine maybe common ancestors and say something about evolution of not only the molecules, but also the organisms that possess them. Okay, so this was pretty interesting advancement for the time being, because actually at that time there was not enough data. We didn't have enough data. Actually, sequencing technologies were more advanced actually for amino acids than nucleotides. And something that was very interesting that I still think is cool to have as a kind of foreshadowing of what was going to happen next is the requirements that they proposed at that time. They said, okay, if we really want to understand molecular evolution, we really need a few things. We need larger number of homologous sequences, right? So we need to have statistics. We need to have data, and for that, kind of technologies like sequencing technologies came and changed the history, right, of molecular evolution. They also said, well, it's important to consider the genetic code. The genetic code is important, and they didn't give any like specific details, but they said, okay, this we need to understand it well in order to kind of know kind of evolutionary change. And finally, the thing that actually catch my attention when I was reading these papers was they were already interested in the idea of studying the 3D structure of molecules in connection with molecular evolution. So the point they wanted to make about kind of using molecules as ways to understand kind of evolutionary relationships was the idea that evolutionary change, or let's say fixation in molecules, might have some sort of a constant rate, kind of like rings in a tree, ticks in a clock. So in this case, I'm showing you some islands in Hawaii that have different ages, and here I think Hawaii is the oldest island, and Hawaii is the newest island with a little bit less than 500,000 years of emergence. And then when people start looking at kind of genes of fruit flies and birds, then they compared what was kind of a metric of evolutionary change. So basically, distance between the same genes, and then they were able to determine kind of a linear relationship between kind of time of emergence of these islands and actually the number of changes that these genes happen, right? And this was kind of a nice observation. Is this idea of Pauling and Zuckerkandl, that maybe if we look at kind of the fossil record, if we look at the molecules from different organisms, maybe we can actually use it as a metric of evolutionary separation, right? So this is something that has been around for many years. It has been useful because it appears that there's a constant rate of molecular change, and then if we use that, maybe we can build a better history of biological lineages. And obviously, in this seminar, in its audience, this is kind of bread and butter, but still, to me, it's still a very interesting observation that kind of has led to multiple new models of evolution. Now, the issue is, and we like things to be linear, we like things to be kind of simple and predictable, but what happens in reality is that rates are not really constant, right? So we do have bursts of amino acid changes in proteins, and many times those are related to kind of acquisition of new functions. Then we also have pseudogenes that allow many more substitutions. And finally, and interesting to me is that there's a strong influence on the tree structure of the protein. So different sites of the protein, different kind of positions in the 3D fold will have different kind of rates for change, and that's not necessarily accounted by this simplified view of the molecular clock. Okay, so for that, people decided, okay, well, why don't we start building models of sequence evolution? I'm going to focus in this talk on models of amino acid evolution, where you have a sequence, and then you want to kind of determine the rules of change, and then see if that coincides with what we observe in nature. And then one of the earliest models by Kimura proposed that kind of most mutations are neutral; they're the result of genetic drift. That kind of led to this idea that maybe all the mutations are neutral, but then these days it's been kind of accepted that although many mutations are neutral, obviously there are others that are not, and it's more like a mixture of changes that we experience and that molecules experience. Okay, but then in these early models of evolution, well, people thought, well, at each side, at each position, I'm going to have a Poissonian distribution of the substitution rates. Okay, so at each side I have a Poissonian distribution, and then I assume that each position has kind of independence. Each position can change by itself, but the distribution is Poissonian. However, this view is not complete, because what we really observe in real data is a phenomenon called overdispersion, where the variance of the rates tends to be higher than the mean of the rates. And if we had like an independence assumption and a Poissonian distribution, then that we expect them to be the same. So in real data, we do have this overdispersion, and it was important to kind of update models to account for that. And one way to account for that is to kind of realize that the rates are not uniform. So they're not the same. So the rates are changing across the whole protein. And models like the RAS model it's called rates across sites were introduced, and then that led to actually a gamma distribution of rates across the protein, still constant but different across the different sites, right? And that led to a kind of more realistic representation of how rates in protein evolution happen, but still they assume that each rate is constant. Later, there were studies that show that even within a single site, even within the same site, there's changes across taxonomic groups. So not only we have different rates and different rates across sites, each site could also have different rates as the taxonomic distribution of organisms happen. So there's a complex relationship of all the changes in sequences, and ideally we would like to account for all these properties. Okay, so there was also this idea that sites might change together, this idea of covariance in sites, and then people thought that maybe accounting for those changes, those kind of correlated changes or correlated mutations, could maybe tell us something about kind of what we observe in evolution. And when we include this in our models by design, then we see that we have now gamma distribution of rates and also heterotachy, this change across different clades. So this was interesting because it's telling us that kind of connected changes are important, or epistasis is important for the observation of this variability in rates. Okay, when people then start thinking about structures, when maybe we make decisions on the rates of change based on the structure of the proteins, and then if we let these rates of mutations, rather than being a fixed parameter, we leave it as a free parameter, then people start seeing that overdispersion happen in the sequences that were generated with this model. So little by little, more and more kind of features of real data were captured by understanding important concepts, but most of the times these were kind of done by design. Once we understand the concept, then we actually add it as a feature of our model. Studies by Pollock also developed kind of a model that was inspired by thermodynamics. So basically, the mutation probability look at compensatory changes, and then that actually allowed these models to kind of display a phenomenon called kind of like an evolutionary Stokes shift, where now changes at different times in the evolution of the change had different kind of energetic responses, and going back and forth between one change was not identical. So that also accounts a little bit for the idea of coevolution in proteins, and those actually require the knowledge of the tree structure of the protein. Okay, so these are kind of this a little bit of a recap of several models of evolution that have been trying to kind of realistically represent change in proteins. And from our side, from the side of my lab and the work we've done throughout the years, we've been interested in kind of studying the statistics of protein families. We're interested in looking at alignments of families, and then kind of identifying features that are connected with the idea of covariance or coevolution. So the way we've been working with this protein families is by kind of modeling the joint probability distribution I'm showing here of whole protein families. We start as an input, we have multiple sequence alignment, and then we essentially model kind of pairwise interactions and local fields and their distributions. And the idea is to identify how sites across these multiple sequence alignments are coupled. And we're interested in obtaining those parameters that we call kind of couplings that are directly connecting two sites in a protein family, right? So in this example, I'm showing two positions that are strongly coupled, and some observations we had years ago was that these positions tend to be close by in the 3D structure of the protein, while the ones that are less coupled, they tend to be kind of separated in 3D structure. So this actually was very interesting observation a few years ago, because this basically told us that if we have enough information of kind of coevolution across sites, then maybe we could say something about contacts in 3D space, long-range contacts, and then this led us to predict structures and predict complexes and predict the effect of mutations. Now with AlphaFold, then kind of a similar concept is integrated in these large language models that predict structure, and this is one idea that comes directly from the understanding of folding and evolution. Okay, so but what is also interesting is that the collection of these parameters, so this is kind of a Boltzmann distribution of this joint probability, the collection actually represents some sort of a fitness score for the whole protein, right? So because this is a global model, this global model is basically built with this collection of couplings across all the different pairs of sites, then an effect or a change in one position will have a direct effect on all its strongest coupled pairs, right? It will have an effect on all of them, but especially in those that have the strongest connections, right? And this phenomenon, or this scoring that we call the Hamiltonian of this probability distribution, actually turns out to be a proxy for fitness, and that's something that we are going to be using to actually create forward models of evolution rather than just understanding the structures of proteins, right? Okay, so how can we use this to kind of build a new model of evolution? Well, for instance, we have the motivation that maybe because this is a global model and it accounts for large collection of sequences and their changes, we motivate this by saying that maybe this is going to capture evolutionary pressures for not only function but also folding. And then structure, folding, and function, they're all obviously interconnected because those are features that we want to preserve through the course of evolution. Now, if we are able to infer these restrictions, and then if we use those restrictions in an evolutionary model, maybe we could actually represent this dynamics of change in a kind of realistic way. That's kind of the main motivation, that because we have these probabilities that actually kind of represent change across all the sequence in a context-dependent manner, maybe this is a good way to kind of decide or at least probabilistically, stochastically determine the probability of change in a sequence. Okay, so then the idea is kind of a DCA Hamiltonian maybe captures the environment of this family, and obviously the definition of a family is a little bit subjective, because first we need to make a collection of sequences that are related, But still, since also there's been a lot of advancements in creating this kind of family definitions, our hope is that maybe this will help us kind of encompass all these properties that I described before for other models into one simplified model. Okay, so this model we call it sequence evolution with epistatic contributions. So the idea is that, or main point is that these coupling terms that are learned from the family are going to be very important to determine kind of change across the evolution trajectory of this protein. So we start by basically learning the quote-unquote fitness of this family. So we have our multiple sequence alignment. Then we, using direct coupling analysis, we actually estimate this joint probability distribution, and then what it means is that we kind of learn this EIJs and local field parameters. Now we propose a way to do kind of the estimation of parameters that is efficient, but there's multiple versions of this kind of solution for these parameters. And in this case, we're going to focus only on a couple, but that's kind of a topic of research, people trying different ways to estimate parameters in a kind of more unique way, because the actual exact parameter estimation is intractable, is a complex problem. Okay, so once we have these parameters, then we take kind of a source sequence, then we uniformly just randomly select one position for change, but then we make a decision if we want to make changes based on a conditional probability. So if we pick site I, then the probability of change will depend on its context. So basically, it will depend on the rest of the sequence and what are the couplings, the sum of couplings, and then the local fields that this new change is going to bring. So if the change is going to be very deleterious, probably this is going to show up as a smaller probability of change. While if it's a conservative or neutral change, it might have a larger probability. So we keep doing this. We select and change depending on these conditionals, and then we can represent kind of this evolution trajectory by quantifying this Hamiltonian, right? Each sequence will have its own age or Hamiltonian, and we're using it as a way to track kind of the evolution of the trajectory, this protein. So this is one example when we run one of these simulations of evolution. Then what we see is that this kind of fitness proxy is changing, right? It goes up and down, and as we have changes that are deleterious, then often we see maybe compensatory changes or epistatic changes that brings things down to a more favorable state. So typically a more negative Hamiltonian means more favorable, kind of like resembling an energy. There's this analogy of energies. But what is interesting is that we observe that even though there's ups and downs, we do maintain some stability. So we do maintain stability on the energetics or fitness, which is kind of for us kind of a proxy of kind of neutral evolution. So we're trying to maintain certain kind of fitness across many changes. So in this case, I'm showing you two examples of two proteins and two protein families. So the LAC-I family and also the transcription factor CPXR family. And here we see how kind of these trajectories after 5,000 evolutionary steps actually change the fitness, but then at the end they maintain some sort of a mean value that is consistent with kind of neutral evolution. Okay, and here a note I mentioned about estimating parameters. So there's a few ways to estimate parameters, and then here we focus on kind of one way to estimate parameters called the Boltzmann machine DCA. In this case, this is kind of a variant of DCA that uses sampling to kind of fit the sequence statistics of the model. And the consequence is that we have better representation of the initial statistics of the MSA, as opposed to another kind of way to compute the parameters that we've developed called the mean field DCA, where it also learns property parameters, but it's less accurate in resembling the statistics of the initial input set. So what we did is in most of the experiments, we try using both of these parameter estimations, and we realized that statistically they're all equivalent, but BMDCA is better at capturing the initial statistics. Therefore, most of this analysis we kept BMDCA.
Dr. Morcos, can you just slide back to the definition of your Hamiltonian there?
Yeah, of course.
So most of the people on this who watch this seminar series, including myself, are more familiar. I mean, I think of this as sort of being like a mutation selection model. But you phrased it as a little bit differently, and we're talking about different means of making estimates for this type of model. Is there any way you can sort of give some intuition about for people who are familiar with like more molecular evolution phrasing? How can we imagine this?
Yeah. So is your question about kind of what is the difference between?
Well, so right. So am I correct in thinking that we sort of have you're introducing mutations and they sort of get accepted and rejected according to this like middle probability?
That's correct.
Okay, I'm happy with that. But okay, connecting that with the Hamiltonian, is that you're looking at ratios of Hamiltonians, and then that's sort of your probability of acceptance?
Yeah, so basically, yeah, I'm glad you asked this. So there's slightly different things. So the probability of acceptance, the selection probability, is a conditional probability. That also depends on this collection of couplings and local fields, but it really looks at a change at one position and then its context, the rest of the sequence. And that's going to determine the probability. Then the Hamiltonian here, this plot that I'm showing the trajectory, this is basically this probability here, which is not a conditional, it's a joint probability of the new sequence, right? So as we make a change, then there's going to be kind of a new probability that we can score. And then essentially we have a probability, but then here we decide just to basically plot this H, which is completely directly related, right? So this H is the one that we are plotting rather than the probability. But it's slightly different. The probability of acceptance is it's a conditional probability, and what I'm showing here, the Hamiltonian, is more like the energy. It's an energy of the sequence.
Right. Okay, that's very helpful. And these methods of estimation, I mean, is there a way to give intuition of the? I mean, for me, I would think, okay, we're going to do some sort of maximum likelihood fit, but I'm guessing maybe that's intractable, or that is the what you do, but you're just using
direct coupling analysis. Yeah, exactly. So here the way we estimate is by modeling this joint probability and then doing maximum entropy maximization, and then this EIJs, at least one in one of the estimates, is we basically invert the cross correlation matrix of the frequencies, the pairwise frequencies. That inversion actually shows that it's a good approximation for these parameters. But then I also mentioned that there's like a more involved way, more complex way of basically just doing sampling and then trying to get EIJs kind of the old-fashioned way.
But still, the objective is a maximum likelihood type objective, but you have some smart ways of getting there. Is that true?
Yeah, exactly. So you could see, yeah, there's also a version of this that uses maximum likelihood, like pseudo likelihood optimization. And yeah, there's multiple ways, and one is like the one that I describe here is either you do kind of a mean field approximation, or the other one is you just do like sampling, and then you make sure that you agree with the parameters. But then maximum likelihood could be another way. Yeah.
Okay. Thank you so much. I appreciate it.
No problem. Yeah. Please stop me if there's more questions from the audience. Yeah. So I think I agree that this comes from like a different angle as a traditional kind of mutation selection kind of field, this comes more from the modeling of the folding and its connections, and then we're trying to kind of reshape it into one of these mutation selection models. Right? So maybe the kind of the jargon is slightly different. Okay, but the point was that we wanted to check against these previous models, and we wanted to see if their consistency, if this model, which is very simple, but once that you have this joint probability and it's conditional, there's really nothing more than we do. We just mutate and select. Something interesting that we observe as a consequence, more than as a design, is we do observe this over-dispersion, this idea that the variance of the rates at each position is higher than the mean. So it's more than one. So this is something that is being observed in real data, and then that models that only assume Poissonian distributions don't get. But here we do have this for this example of two families, and we have it for many others. I'm showing that, yeah, this over-dispersion RT more than one happens. And we also observe that kind of the rates, the distribution of rates, in this case, the rate is defined as the ratio of this kind of synonymous substitution divided by non-synonymous substitution mean times. We observe also kind of a distribution of different rates that resemble kind of a gamma distribution. So rather than imposing this gamma distribution, this is basically a consequence of this kind of mutation selection procedure, right? So we do have our expression. We have gamma-like distribution of rates, and we also observe this phenomenon of kind of evolutionary Stokes shift. So here what I'm showing in these plots is I'm showing kind of different sites where I have my change in Hamiltonian. Remember, this H, we use it as a proxy of fitness or energetics. So when I have a forward mutation, I will have a delta in my Hamiltonian. And then when I have a back mutation, so reversion, then I will have also kind of a change in my Hamiltonian, right? So after only a few steps, only a few changes, typically I do have like a linear relationship. There's not a lot of distinction between going forward and backwards in terms of this energetics. But then as we progress, in this case, as we reach 1500 steps, then what happens is that now I'm acquiring other changes across sites that are compatible or epistatic with respect to the initial changes. And now we start deviating from this diagonal, where now a forward change is not necessarily one-to-one with a backward change. So now we start seeing this kind of shift that kind of it has been observed to be important kind of proxy of coevolution. Right? So since we knew originally that this direct coupling analysis captures coevolutionary relationships, this was kind of expected, but still it's interesting that appeared as a kind of consequence in this model. Okay. So in other words, if we have different energetic consequences at different stages in the dynamics of the model, this is kind of a good way to say that this evolution could show entrenchment and contingency, these are important concepts that kind of a proper evolution model should have. Okay. Other way to see it is that reversibility is not generally possible after many changes, because there's been kind of compensatory changes that are dependent on the context. And then after those changes happen, then you get your sites get entrenched, and then the energetic change is going to be much more restricted. Okay, and as I mentioned before, this is something that was observed by Pollock et al. when they incorporated a kind of a thermodynamic model that accounts for these changes at the kind of structural level. and this is coming directly from sequence composition, and that does not include like a explicit representation of the structure. Okay, so as a summary of the statistics of the model is that we're able to kind of have this kind of stable energetics that we kind of connect to the idea of neutral evolution. You want to maintain certain fitness because this is not a model of neo-functionalization, is a model of more like neutral evolution. But we do observe over dispersion. We do observe different rates across sites. And interestingly, we also have this evolution Stokes shifts. We also actually observe this other concept of heterotoky, that different sites have different degrees of heterotoky. They go and across more kind of clades. When you check at different kind of subgroups of your kind of evolutionary trajectories, then we do observe different rates at different times. So kind of or take away a message was that by capturing this kind of constraints that are kind of obtained with these direct couplings, then doing this simple mutational selection process informed by this conditional probability, we get all these statistical properties of previous models all together, right? And without the need of maybe structure, because it is all captured in the kind of MSA. But very interesting or important for us was to be able to kind of test these models in the lab, because maybe obtaining proper statistics is an interesting theoretical advancement, but we always want to make a connection with kind of the bench with kind of experimental observables. So what we did is we decided to basically take one single family, the TEM1 antibiotic resistance family, and then we decided to take one of those proteins, evolve it using this SEEC model, then synthesize some of the variants, transform them, and then check if they would confer growth in E. coli, right? So the idea is that if this evolutionary model captures kind of realistic changes, it probably will lead to sequences that are also functional. And if the sequences are not functional, then we will not observe growth. So this assay, it's a perfect assay for these models. So what we can do is we take TEM1, we evolve it, then we look at the trajectory, and what we thought was, well, let's check some variants at the beginning of the trajectory, then sometime in the middle and late, just to see kind of what is the effect of introducing many changes, and see if kind of these changes in fitness according to this Hamiltonian made some sense, or they were kind of predictive of also fitness in the lab. So we started by doing these transformations for beta-lactamases, and then we used antibiotic concentrations that still were kind of high, but not increasing too much the evolutionary pressure. So in this case, we use 50 micrograms per milliliter, and then we sample from this, let's say, 3 areas of the trajectory, and then what we checked was, okay, do we have growth? So this I'm showing basically kind of the beginning variants, 6 of them, then the middle variants, and then the late variants. So late variants accumulate, I think, up to 47, maybe 47 mutations with respect to the original. So even though we introduced 47 mutations, we were kind of happy to see that this late 1, 2, and 3, all of them grew well relative to the wild type. They were very similar, and the empty vector control obviously didn't grow. And we see it also for the beginning and middle variants. So essentially, all these variants that were evolved with this method actually were functional. They fold and they confer resistance. which we thought was very interesting. And it was some sort of a way to kind of confirm the validity maybe of this model. But something that is very important to mention is that for this to actually work in the lab, looking at the genetic code, kind of what Linus Pauling and Zucker Kendall mentioned, was very important. Because in our initial model, we were able to actually accept mutations that could have more than one change at the codon level. So what we did here, and then when we do that, then we accept changes that are kind of less probable because they might have even more than 2 or 3 changes at the nucleotide level. So when we were considering that initial model, we were not having this same results. We were having maybe at the beginning things were okay, but then we would not see growth. But then when we restricted that the changes are only accepted if they are also within one change at the column level, then that's when we started seeing this interesting and growth. Okay, so that was like a key observation that is important for this model. Was important for this model. Okay, so then we said, well, it looks like everything grows, so which is nice, but it's also pushing us to say, why don't we actually try larger kind of selection pressure, so larger antibiotic concentrations to see if things still kind of provide resistance. And something interesting that we observed in this case, rather than just growth, I'm showing you the relative fold growth with respect to the wild type. So wild type is 1. And then we see something very interesting, that most of the variants that we produce with the model actually perform better than the wild type. So most of them actually grew much better with respect to the wild type, even, and curiously, even at the latest stage where we have more changes that are acquired. Just want to note that this changes, this evolution trajectory was done with the mean field approximation. The estimation was done with this mean field kind of inversion of the correlation matrix. And then we also tried with the Boltzmann machine DCA, the one that infers better the statistics of the family. And curiously enough, when we use the BMDCA, then the relative growth is not as good with respect to wild type. So we still have variants that grow at higher kind of amino acid concentrations, but we don't have exactly the same behavior as with mean field. What is interesting is that, let's say in this case where we have consecutive variants 1, 2, and 3 that actually go up in terms of the Hamiltonian. So when we have like a unfavorable fitness, then we do observe kind of a decrease in kind of a function, and then a lower performance when we kind of check for growth. What we realize is that BMDCA, as it's trying to really capture the statistics of the whole family, it's really accepting changes that are maybe going outside of the context of E. coli. So essentially it's accepting changes for the whole family, and that is not necessarily ideal for the local context of E. coli. That's so far our interpretation why maybe BMDCA is not performing as well as with the mean field variants, where everything grows better than the wild type. Okay, and finally we look at the structures, and then we wanted to understand, okay, where do we have the changes? Where are the places where we accept changes, and where are the places that are more restricted, given that there's no kind of implicit restriction imposed by the model. And here we're having 2, mean field and Boltzmann machine DCA. As you can see, the BMDCA has many more changes, or more changes than the mean field, and that probably explains why maybe the the performance was not maybe as good as this one. But the note here that I wanted to kind of highlight is that if you look at the binding site, the binding sites is almost untouched. There's only very few changes that are kind of imposed in the binding site, which makes sense, right? Because those are the regions that are highly conserved, and in order to preserve function, they should have lower probability of change. And that was like also a nice and interesting observation of the model that accounts for those conserved regions as a consequence rather than as this design. Then we also found another region that was mostly untouched, and then that we realized that was a region where we have chaperone binding. And we thought that was also interesting, that it's also preserving regions that are important for molecular interactions, for protein interactions. You don't want to change much those ones unless you have some sort of a coevolution with the other protein. But at least in this case, the model kept that untouched. Okay, so it is not only that we're changing the surface, as in many models we observe that only surface changes are allowed. Here we do have changes across the protein, but some key areas are restricted, and that makes sense. Okay, so I think I'm almost out of time. So I just wanted to summarize kind of what I discussed today. So basically, by learning from the sequences, by learning this joint probability distribution, and then having this conditional probability at this mutation selection stage, this evolutionary model actually captures many properties that have been observed in real data, and that includes kind of like the union of many models that have been developed through time. And I'm sure there are many other models that might do similar things, but at least the ones that we try to understand are compatible with this model. And these properties that are important statistically are this kind of that the models have variable rates across sites, the observation of over dispersion, heterotachy, this evolution Stokes shift that is connected to coevolution and entrenchment and contingency. But then this model also actually leads to sequences in the lab that are functional. So this is like in our opinion, a rigorous test that these models really can achieve kind of evolve sequences that are not going to be disrupted by kind of folding kind of requirements or functional requirements. And as an interesting kind of corollary or observation was that somehow this variance conferred even higher resistance than the wild type of E. coli. And our interpretation is that we're really obtaining kind of high probability changes across the whole family. So maybe that combination leads to kind of a more stable or functional protein. And we think this could be a useful approximation for kind of evolution of protein families. Maybe there's a way that these models can be kind of adapted or introduced, maybe in tree construction, which maybe that's something that people in this seminar might be interested in, as every phylogenetic tree construction has its own inherent evolution model. Maybe this also could be used in some way to do ancestral reconstruction, maybe forecasting and design. So yeah, with that, I would like to just finish by acknowledging people in the lab working on this: Charisse Narti, Sofia Alvarez, and Alberto de la Paz, who were working this project throughout these years, and then funding agencies for supporting our work. And thank you for listening to this seminar.
Awesome. Yeah, thank you very much. Yeah, I mean, it is really exciting when you can do something computational and then it works in the lab. That's really amazing. Can we actually dig into that a little bit more? I confess to not really understand the difference between BMDCA and mean field enough to really follow why it is that the mean field worked significantly better in terms of like the prediction of
Yeah, so I think so. we're still actually trying to understand exactly why. We have now better clues. So basically, the way the parameters are inferred is slightly different, and the mean field version really focuses a lot on kind of the strong couplings. So essentially, it highlights more the strongest connections, and then it dilutes more the weaker connections, while that could lead, when we think about a kind of a generative property, so if you do sampling, then that leads to sequences that are not necessarily exactly as in the whole family, while the BMDCA enforces that. It tries to maintain also kind of the weaker couplings and the weaker statistics, with the hope that maybe the sites, the pairwise statistics are preserved. So they're similar, but with slightly different consequences. And from the point of view of this kind of mutation selection procedure, it means that the BMDCA allows more changes, because the probabilities are slightly different. The lower probabilities, I would say, are more similar to the ones that have high probabilities. So the mean field DCA is making a little bit more stark differences. But what we see, and that's something we're working right now, is we saw, and I'm sorry I didn't give enough details here, but if you notice in the y-axis here, we have a T, kind of a temperature, a selection temperature that can be adjusted. So so far, I think here we use a temperature of 1, but what we notice is that if we play with that temperature, so basically if we decrease or increase the temperature, so then we can, with BMDCA, which basically confers the whole statistics of the family, we could also play a little bit with the selection parameters. So for higher, let's say, selection pressures, maybe we can reduce the temperature, and then that will confer maybe less changes in the sequences. And so far, our preliminary results show that it could behave very similar to the mean field DCA. We think that's basically kind of the solution to that problem, that we fine tune that temperature, so we allow like higher expansion or less expansion to kind of new parts of the family.
Right? Okay, but I mean, I think you were even just touching on this. I mean, so you were talking about going outside the E. coli context, and so what you're really saying is like how much entropy are you trying to introduce into, or not entropy in this? I mean, like how much sort of randomness are you allowing in the variants that you're proposing?
Yeah, that's correct. Yeah. So and even in this context, I wouldn't even say randomness, because maybe those changes are still coupled and they're important, and they have been observed in the family, but maybe not in the context, the local context. So I feel that's the reason. To be honest, we have not completely characterized this, so that's something we're working right now. But because we've done these experiments where we kind of, like you said, reduce the entropy, and then we've seen that now things start to behave a little bit more like the mean field, we think that's kind of the solution, right?
Cool.
Like exploration of the family, it's fine-tunable. Let's put it that way. Yeah.
I mean, this is maybe just like a silly computational biologist looking at lab experiments sort of way, but I do wonder, like, do you have to do a growth assay, or is there another assay that you could do that would be like an easy, like a more high throughput proxy? I mean, you talked about stability. Like, could you do a high throughput stability assay to get a first pass on these proteins before you actually do the growth assay?
It's possible. Maybe stability could be good, but somehow we thought that, because in theory, what we want to preserve is kind of functional outcome, or like a physiological output. We
Right.
thought that the key is that this would confer resistance, right? Because we might have higher maybe stabilities, but that maybe not necessarily will confirm kind of survival.
Right. No, absolutely. I mean, the growth assay is the gold standard. Absolutely.
Yeah.
I was just sort of thinking if there was some like
a
intermediate
high throughput way.
Well, just a way that you could sort of filter out some of the sequences to get even better performance out of the actual growth assay. Mm-hmm. But I am a little bit curious, like, it's sort of amazing, right? You have this, what was the beginning sequence that you started with for the E. coli?
So we have the wild type, and then beginning, I think it has only like a few, maybe 2 or 3 changes.
But like, why is that wild type sequence not optimal? I mean, you did your computational evolution and then it got better.
Yeah, so you mean that basically wild type? Why is it? I think this is because look, the antibiotic concentration was much higher.
No, but going back to the 50 milligrams, I mean, like like after evolving your protein, like computationally, it has better performance, right? Am I missing something?
Correct. Yeah, I think so. So very comparable at 50, but yeah, in many cases better than the wild type. Yeah.
But that just seems surprising, right? Like this, this beta lactamase has been evolving for millions of years. Like why is it not more optimal?
Yeah. So to be honest, I was happy to see growth, right? And this was kind of a little bit of a kind of a surprise that we're getting kind of higher performance. I feel that this is connected with this idea of evolution doing what it needs, right? So you have an enzyme that just needs to work in order to survive, but maybe there's variants across the family that maybe have optimized kind of resistance kind of features. And here this model, because it's borrowing from what's been accepted also in other members of the family, maybe it's some sort of finding this optimized enzyme that whose features maybe were important in other members of the family, but here we're kind of putting them all together. That's kind of the interpretation that I have. But my expectation was to have a little bit either the same or even lower growth, but somehow, yeah, we're doing some sort of a maybe optimization. Maybe there's in the target TEM1 in the source, I'm sorry, maybe there's things that are kind of in the middle of evolution, but they don't need to change yet to be optimal. But yeah, this is a good question. I'm not sure if we still know what's the answer. Yeah.
Oh, that's really exciting. So you said as far as next steps, you're just continuing to look into more details of how this all works. Any other next steps that you're thinking about?
Yeah, so what we're also doing, and this is a separate project in the lab, is we're looking at the idea of understanding kind of the families, not only through their extant sequences, but also through kind of a model that generates new sequences. In this case, you generate new sequences through a model of evolution and dynamics, but then we're also looking at ways to kind of generate sequences through machine learning, right? If you learn features, these latent spaces, and then can we create kind of a latent space for the whole family, and then see how these evolutions actually track in this space. So kind of combining a couple of projects to understand the evolution of sequences. That's one thing. The other thing is this idea of the selection temperature, because we think there's this whole theoretical background behind the idea of the selection temperature, and here we're really seeing a connection in the lab, and that's something we also want to dig a little bit more.
Cool. Well, that's been a really neat talk. This is a great place to start our trio of 3 talks. Mm-hmm. And thanks for your amazing talk.
Well, thanks a lot for the invitation. I'm really glad I had the chance to present, and I'm looking forward for the other talks. For sure, I'm gonna join and watch them.
All right. Thank you, Faruck.
Thanks a lot for the invitation, Eric.
Bye.
Goodbye.